everybody, welcome to Chapter 8 on cell-mediated immunity. You guys are going to love this chapter because most of it you've already heard. So all of the really difficult, again, functional stuff, how all the stuff works, we have covered in previous chapters. So mostly this is going to be an overview. So sit back, relax, and start putting things together.
Hey y'all, this is Dr. Ali, and we're going to keep going on with our cell-mediated immunity. Recall with cell-mediated immunity that we are in particular dealing with intracellular infections. Now we have two types of intracellular pathogens. We have those that are considered to be facultative, and these pathogens don't necessarily need to be within a cell to survive, but rather have adapted some sort of evasive mechanism that they can themselves avoid destruction and remain happy and healthy within the cells. We also have our other category of pathogens that are obligate intracellular, meaning that they don't have any choice but to replicate within inside the cell. And a great example of that are going to be our viruses. Recall from Chapter 6 that whenever we have an intracellular infection, we really are going to engage the Th1 subset of cells. So again, there are a few terms that I always think of being very directly related to one another. So if I have an intracellular infection, I'm pretty much going for the fact that I'm having a Th1 response. My Th1 response makes me immediately think that I'm going to induce cell-mediated immunity. Why? Well, cell-mediated immunity is how we destroy cells that are intracellularly infected. So it's sort of that train of thought. And again, sometimes it's going to save you a lot of time when you are dealing with a question, trying to determine if it's a Th1, a Th2, or either a Th17. Recall that cell-mediated immunity is going to be dictated by a Th1 response. The three cells really that I want you to focus on with a Th1 response are going to be our cytotoxic T lymphocytes as well as our macrophage. And we're going to go through each of those cells and show you how Th1s are going to affect their behavior. It's probably also good to remind yourself, if we go through these, what cytokines play a major role. And in a Th1 response, it is always going to be our interferon gamma, which is that incredible pro-inflammatory cytokine.
The Th1 response activates macrophages, and I want you to think about macrophages mostly, even though B cells can become activated as well, by way of the cytokine interferon gamma. Because I really want you to think about this Th1 response as our response against intracellular pathogens, okay? Now, yes, B cells become activated and interferon gamma will help induce class switching of B cells to IgG, but as far as the response in general, I want you to predominantly think about the macrophage, okay? And it is really important that you understand that the major cytokine for macrophage activation is interferon gamma. So once these macrophages are activated, they upregulate their MHC, they upregulate all of their oxygen-dependent, oxygen-independent radicals, and they are able to then eliminate these intracellular pathogens. And as we said before, for the B cells, can help the B cells class switch to make IgG antibodies. And the IgG antibodies is no sort of random thing. It's not, like, oh, we'll make IgG. Because those IgG antibodies, remember, are the only antibodies that can opsonize, and they can help assist the macrophages with phagocytosis. The IgG, and remember the other really important opsonin is C3b, those opsonins help to enhance the process of phagocytosis and make it about 4,000 times more efficient than it is by itself without the help of antibodies and complement.
When we're talking about cell-mediated immunity, we really do like to focus on the cells that are going to be directly involved with the removal of either intracellularly infected cells or tumor cells. But we would be completely remiss not to mention the role of antibodies in this overall process. Recall that our Th1 cells are going to have upregulated on their surface the CD40 ligand that is going to serve as the co-stimulatory molecule for not only our macrophage but also our B cells. Now, when you add to that mix the interferon gamma, the following will result. Our B cells are going to undergo activation, and we will have B cells that are going to undergo isotype switching. That isotype switch will primarily be to IgG, and the IgG then can be utilized using a variety of different approaches. It can lead to increased activation of the classical complement pathway. The antibodies can also serve as opsonins, and you can even use these IgG antibodies to initiate the antibody-dependent cellular cytotoxicity. Now, here is some serious review that we're going to very briefly go through. Again, I just want to remind you that this is sort of a two-way street. We have crosstalk between our antigen-presenting cell, here is the macrophage, and the T cell. Once the T cell becomes activated and differentiates into a helper T cell, it is then going to be able in turn to help activate our macrophage. Again, in a very similar manner as what we saw with activation of our B cells. The helper T cell will express high levels of CD40 ligand on its surface, and this is going to provide that co-stimulatory signal necessary to hyperactivate the macrophage. And again, you will have a pretty significant influence from the cytokines that are being produced by that helper T cell. Now let's really go back and remember all of the different aspects of this interaction with our macrophage. Please recall that our macrophage are going to recognize the pathogen. They are going to phagocyze the pathogen, and they are also going to start secreting their own set of cytokines, including the endogenous pyrogens, the chemokine, IL-8, and also IL-12. And it is the IL-12 that is going to start the differentiation of your CD4 positive T cells into Th1s. And remember, once we activate a cell and it becomes a Th1 cell, it will respond by producing very, very high levels of interferon. So I think it's fairly simple to say that the interplay between the IL-12 and the interferon gamma, they're going to work to really promote a very strong Th1 response. Now, there is, again, remember when I started this, I always said, when I hear I have an intracellular infection, I know I'm going to clear it by cell-mediated immunity. I also said that every time I hear cell-mediated immunity, I think that I'm having a Th1 response. Again, all of those go hand in hand. Now, before I get Dr. Moscatello in here to talk to you about CD8 positive T cells, I just want to add one other aspect to this. When we get into our type 4 hypersensitivities or delayed type hypersensitivities, we are going to find that they are also produced by a Th1 response. So whenever I hear delayed type hypersensitivity is intact, I know that my Th1 cells are working just fine. So again, write yourself a note. Intracellular is going to be cleared by cell-mediated immunity. Cell-mediated immunity is going to be a Th1 response, and I can directly affect delayed type hypersensitivity through Th1. If you know that, you're going to be really solid.
All right, Dr. Muscatello again. Hoping you recall this figure because we've gone through it a few times now. And it's really no different when we're talking about cell-mediated immunity than really when we're talking about humoral immunity. Because the entire goal, again, is to bring the antigen and the cells together in the secondary lymphoid tissue to generate effector cells, which then get back into circulation to help us clear up the infection. Once they get released into circulation, they go back to the site of the infection again because they're getting recruited there. Now, the only difference in what we talked about in this chapter versus some of the other chapters is that the effector cells are T cells that we've generated here. Dr. Alley just talked to you about the functions of the T helper cells. So we know that those effector cells are the cells that are going to produce cytokines. They're going to differentiate into subsets to sort of direct all aspects of the immune response. Now we are going to talk about the other effector cells, which are the cytotoxic T cells. Now, during an immune response that requires T cells, the proliferation of these T cells is really sort of in this massive autocrine fashion. And the way I always sort of describe this is like it's like a political commercial that we're always so tired of seeing during election years when we're talking about IL-2, right? So IL-2 is made for T cells by T cells, okay? So when T cells become activated, they make IL-2, and that IL-2 then they utilize to help proliferate, help activate, and then generate all of these immune responses that are reliant on T cells. Remember, T cells really control the entire immune response. So they are required for class switching and for a really good cytotoxic response. And the most important cytokine for that is going to be IL-2. Now, if we're talking about a Th1 response, T cells also can provide interferon gamma. And remember, interferon gamma is our major cytokine for macrophage activation, okay? And when macrophages are activated, they sort of turn around and help activate more T helper cells. They can get the naive T helper cells once they're activated themselves. Now, macrophages produce another cytokine called IL-12. And IL-12 also helps to not only activate those T helper cells, but allow them to differentiate from the Th0 cell to the Th1 cell, okay? So sort of together, that IL-12 and interferon gamma, if a Th0 cell finds itself in a high concentration of IL-12 and interferon gamma, it knows that we have something that's intracellular or facultative intracellular, and we need to differentiate into a cell that is specialized and capable of handling that particular type of infection. Now, what's really awesome is that we can double dip a little bit here, right? Because we've gone through the Th1 response, hopefully with a fine-tooth comb, and we have also stated that this Th1 response is the exact same response as our delayed type hypersensitivity reaction, okay? We're going to get to that a little bit more in chapter 12. But the mechanism of that reaction, the activation of the macrophage, then activating the T cell giving the macrophage interferon gamma, and or making IL-2 for CD8 positive T cells, that is the mechanism for the delayed type hypersensitivity response. However, what's really important, and we said this a million times and we'll probably say it a million more, this is our normal immune response to, again, intracellular pathogens, but if we're really talking about that macrophage activation, it's specific for those facultative intracellular pathogens. Now, we've mentioned that a million times. Hopefully, you've been through the microbiology, so you know exactly what I'm talking about. But a facultative intracellular pathogen is one that can live inside or outside of a macrophage, let's say. So, for example, tuberculosis. Listeria are really good examples that they get into the macrophage, they get taken up by phagocytosis, but they can avoid killing by a lot of different mechanisms, okay? So this Th1 response is essential for eliminating those types of infections, but also if it becomes activated to things like poison ivy, poison oak, we get the exact same immune response, and now with no pathogen as a target, we simply just get tissue damage. The last of the effector cells we're going to talk about as far as the adaptive immune response are the cytotoxic T cells, or the CTLs. These guys are like the professional hitmen. They go in, they find a target, and they blow it up. So the difference between these cytotoxic T cells and the natural killer cells that we introduced in the chapter on innate immunity is specificity. Cytotoxic T cells, remember, are going to specifically bind to endogenous peptides that are located in the class 1 MHC molecule. Now, cytotoxic T cells are going to leave sort of normal cells alone, so we should not have any T cells, if everything's functioning well, that are cytotoxic that bind to normal self-peptides because those have been eliminated within the thymus. However, if the cell is infected with an intracellular pathogen or perhaps transformed, those T cells that were generated way back in the thymus should be capable now of binding to these different peptides, again, always within the context of a class 1 MHC molecule, and killing those cells. Recall from an earlier chapter, we talked about something called cross-presentation or cross-priming. This is going to be important when we're talking about really a CD8 positive cell becoming a fully effective killer cell. So cytotoxic T cells can be killers without the help of Th1 cells, but they are much more effective when they become fully differentiated active killer cells, which they will do with a little bit of help from the Th1 cell by way of IL-2. Now, I know, because I've been sitting here, that Dr. Alley talked to you about the function of IL-2. She pretty much said she would hunt you down regardless of how much time has passed and ask you what the function of IL-2 is. So I totally expect you to know that. The way I sort of think about IL-2 is that it's sort of like some of these stupid political ads that we see on TV. It's made by T cells for T cells. A lot of these politicians say they're by the people for the people. Yeah, but they're not. But IL-2 is made by T cells, all about the T cells. Recall in cross-priming that an antigen-presenting cell, like a dendritic cell, can engulf an entirely infected cell, and you can actually have now on this APC the expression of class 1 and class 2 MHC against the same types of antigens. So you can have activation of both CD8 positive T cells, and if we drew that in over here, you would actually see a CD4 positive T cell. Now that's going to be important because it's that CD4 positive Th1 cell that is making lots of that IL-2 that is going to help activate that CD8 positive cell to be an effective killer. Now remember, when we went all the way back to the innate immune chapter, we are also going to have production of interferons that may be produced in response to a viral infection that are within this same area of immune cell activation that can increase the expression of MHC molecules. That certainly makes it easier for the cytotoxic T cell to find. So now we're going to talk about how a cytotoxic cell kills a target cell. And you're going to want to listen up here because we're killing two birds with one stone on this one. Because once you understand how a cytotoxic cell kills a target cell, it's pretty much the same for natural killer cells, except for how they initially bind to the target cell. So for cytotoxic cell, the first step is, of course, that we have to attach to the target cell. Now remember, the difference between this and a natural killer cell is that this attachment is specific through the T cell receptor and the class 1 MHC molecule. we get activation, which essentially means that the cytoskeleton rearranges so that the granules sort of face the interface between the target cell and the cytotoxic cell. The granules are then released, and we'll talk about in just a little bit what some of the contents of those granules are. And then the cytotoxic cell, remember it's a professional hitman, it detaches from that target cell and goes on to kill the next cell that might be expressing a foreign peptide. So here we have what we just said in the previous slide in sort of a figure, so it's a little bit easier to understand. So you can see the cytotoxic cell binding to its target cell. Activation of the cytoskeleton moves those granules toward the interface between the killer cell and the target cell, and they release the contents of the granules. Now, one of the important molecules within the granules is something called perforin. So finally, the immune system gets it right. It finally gets a good name, and something was actually named for what it does. So we don't have 8 million alpha betas flying around. We have a molecule called perforin that actually perforates the target cell membrane. Now that would be enough to actually kill the target cell, but remember this is a professional hitman. So in addition to punching holes in the target cell membrane, it's going to pump it full of toxic chemicals like enzymes, granzymes, and cytokines that are going to force this cell to commit cell suicide or apoptosis. Now, if you look on the right, we have sort of a different mechanism of CTL killing. Sometimes I like to refer to this as the A side, which we just went over the hard way, and the B side, sort of the easy way, because sometimes when you least expect it, life is a little bit easier, right? So sometimes, rather than the cytotoxic cell having to completely activate, release granules, all of that mess, because really that takes a little bit of time, it can recognize through a ligand called Fas ligand that there is a molecule being expressed on target cells called Fas. And Fas is generally expressed on the surface of cells that aren't doing too well. It's sort of an indication that the cell may be dying, or at least is in trouble, and those cytotoxic T cells can bind that Fas molecule, and that Fas-Fas ligand interaction is enough to force that cell into apoptosis. That, ladies and gentlemen, is the easy way. Now, again, as Dr. Ellie said, a lot of this is review. We really already talked about natural killer cells for the most part when we went through innate immunity, so here's a nice little summary for you, okay, of the CD markers that natural killer cells are not recognizing antigen through class 1 MHC, but they are actually inhibited from killing if there is class 1 MHC expressed on the surface of a cell. And they detect that through a molecule called HLA-E that we talked about earlier. The effector mechanisms, guess what, are exactly the same as what we just talked about for the cytotoxic T cells. So again, you're going to see the perforin, the granzymes, the cytokines that are toxic and will cause that target cell to enter into apoptosis. So as we said earlier, when we were talking about the innate immune system and the adaptive immune system working together, the antibody-dependent cellular cytotoxicity is really the perfect example of this, because it's a cell-mediated immunity, but it sort of bridges innate immunity from natural killer cells with adaptive immunity because we're actually using antibodies. We always sort of think about this process with natural killer cells, but in actuality, any cell that has an Fc receptor that can recognize the constant region of the heavy chain of IgG, and we talk about that receptor quite a bit, that's CD16. Any cell that expresses CD16 can function in antibody-dependent cellular cytotoxicity. But again, look at the cells here. Natural killer cells, macrophages, neutrophils, eosinophils, all of those cells essentially are innate immune cells, okay? So again, ADCC uses antibodies to help bridge some specificity to the innate immune system. Here's where life gets easy. The mechanism of target cell killing is pretty much what we already looked at. Lytic enzymes, cytokines, and perforin and granzymes. So the ammunition is the same. Essentially, the difference is the recognition of either specific antigen, okay? If the natural killer cell is killing in a truly innate fashion through lack of expression of class 1 MHC, but in the case of ADCC, we actually have an antibody that has to be IgG that's bound to a target cell. That's FC portion binds to an FC receptor on the surface of one of the cells that we just mentioned. So let's take a look at what that looks like. All right, so I know a figure like this can get a little bit overwhelming. So let's just start with the natural killer cell. You can see that you have an antibody, which again has to be IgG, binding to the target cell. Now, what kind of target cell would IgG bind to? Well, it could be a viral infected cell that's expressing viral antigens on the cell surface. Could be a transformed cell. Any sort of molecule that IgG can bind to, meaning that it is non-self, hopefully. IgG binds to that target cell, and you can see the natural killer cell binds to that IgG through that CD16 molecule, which is the Fc gamma receptor, and releases all of those sort of proteins and cytokines that we talked about, perforin and granzymes and other lytic enzymes. Neutrophils, macrophages can do this as well, although they are not known to express perforin or release perforin, but they do release lytic enzymes. So again, they're not going to just whack the target cell like we expect from cytotoxic T cells and natural killer cells, okay, but they can be effective nonetheless. And eosinophils. Now, what's interesting about the eosinophils is, of course, that when we think about eosinophils, we should be thinking about what type of reaction? Yes, thinking about that type 1 hypersensitivity reaction. And ADCC can actually be mediated through IgE in one special case when we're actually looking at a helminth infection.